So for tonight's program, hopefully everybody here tonight took part in the survey, or if you didn't, I hope you're feeling guilty about it, because we're going to hear all the results tonight. And I'd like to thank Phoebe Snyder and Dr. Kaira Wagoner for coming tonight, and I'm going to turn it over to them now. If I can figure out.
Yeah, I just need to share my screen here.
Okay.
There we go. All right, everybody see it?
Yes.
Okay. All right, well, thanks so much for having us. We're really happy to be here, and thanks everyone for participating in the survey. So I have a presentation about, I think it's about 45 minutes that we can go through. And then Phoebe's going to go through the survey results at the end. So does that sound good to everybody? We ready to get started? I wanted to make sure before I start, there's no questions for Jody. All right, everybody.
I think we're good.
All right, everybody's good. Okay, I just want to say too, last time I did a Zoom presentation, my internet cut out and I had to rejoin. So if I go anywhere, Phoebe's going to entertain you till I get back, but I'll just know I'm trying to get back. Hopefully it won't be a problem. All right, so I wanted to tell you a little bit today about honeybee health and our work at UNCG with honeybee health. And I'll start with just an outline of the presentation. I'm going to tell you a little bit background information about honeybee importance and honeybee health, some specifics on honeybee hygiene and chemical brood signaling. And then I'm really excited to tell you about a new tool that we're working on to help combat varroa. I want to tell you about some of my earlier work related to foundation wires in the hive, kind of a summary of the importance of the work, where we plan to go from here. And then I figured, you know, we could do, if there's any burning questions, we can have a little time for questions. And then Phoebe can give the Chatham County survey results. And then I think at the end of that, we can have more time for questions. And of course, I'm always happy to answer any questions by email later on. If anybody wants to talk more, I love talking about bees. So as beekeepers, I think we all know that honeybees are really fascinating creatures. And there's so many different reasons why they're fascinating to beekeepers. And that's also true for biologists. So for biologists, one of the main reasons I think honeybees are so interesting is because of their complex social structure. So we know there are three main castes in the colony. There's the drone, the queen, and the worker. The drone, the main function of the drone is to mate with queens from other hives. The queen, of course, lays the eggs, and the workers complete all the other hive tasks, and there are lots of hive tasks that they're doing. But interestingly, those tasks change as the workers age. So when they're younger, they tend to do things inside the colony, things like brood tending, receiving nectar from those foragers, those older foragers that are doing all the risky business of leaving the colony and bringing back all the goodies. So honeybees are really important because of this kind of plastic behavior. They're important as a biological model. They're really useful for studying behavior, for studying communication. And one of the main reasons for that is because of something called phenotypic plasticity. And what this is you can have one genotype that can become one of two different phenotypes. So what I mean is when you have a fertilized egg and you feed that, you can feed that fertilized egg or that larvae once it hatches two different diets and you get two very different outcomes. So if that larvae is fed royal jelly, you have a faster development and you end up with a queen who can live up to, and this doesn't seem to be true anymore, at least for my colonies, but back in the days before varroa, those queens could live even up to 5 years. Whereas if you feed that same larvae a worker jelly diet, they're going to live a matter of weeks, 3 to 16 weeks, depending on the season. And so that makes honeybees really important for studying things like aging, things like genetics. You know, just with changing the diet, we can get these very different outcomes in terms of aging. So it's a really important biological model for scientists. Of course, we know honeybees are also important as crop pollinators. So they contribute about $16.5 billion every year in the US alone to crop yield. So really, really important. And we know that our plates are much more beautiful, much more delicious with bees than they are without bees. But what about the environment? So there's a study that came out in 2017 that said of the tens of thousands of pollinator species, honeybees account for about 13% of floral visits in natural ecosystems. So we're not talking about crops, but just natural ecosystems. And so that's really a pretty huge amount, 13%, considering that they're just one species of tens of thousands of different pollinators. But the same study actually showed that honeybees fail to visit nearly half of all animal pollinated plant species. They never even show up to those species. So what this tells us is that honeybees are important, yes, but native bees are also really critical to natural ecosystems. And so because honeybees and native bees share resources, that also means they share pesticide exposure, they share parasites. So honeybees are really important also to the environment, not only because of their own natural ecosystem pollination, but because one, they can act kind of like a canary in the coal mine. So, you know, a lot of native bees are not managed, whereas honeybees are managed. So if we see there's a problem with our honeybees, a lot of times that's a good indication that there's also a problem with native bees. And it tells us that controlling honeybee parasites may benefit native bees in addition to honeybee health. So honeybee health, that's my big topic. Is there really a bee apocalypse? And for anybody paying attention, starting about 2006, that was kind of all the news. It hit all the major channels. There were vanishing bees, be afraid, a world without bees was the cover of Time magazine. And the big overarching message was colonies are in collapse. So we heard a lot about colony collapse disorder. Well, a lot of the public doesn't realize that colony collapse disorder is actually a very specific set of symptoms. So in colony collapse disorder, there are very few adults that are found in the hive, but they're not, you know, just dead outside the hive, they're just kind of missing. And then the queen, the brood, the honey, and the pollen are all present, found in the colony. So if we look at the number of hives, this is the number of hives in millions in the US over the past about 120 years or so. And if we look at what's going on compared to where CCD was first reported, you can see that there's clearly something else going on. It's not CCD that's causing this great decline in honeybee colony numbers. So to figure out why that is, we have to do a little history lesson. So the most colonies that we've ever had in this country were during World War II. And the reason for that is that the honey was needed for use by the public. So sugar was sent to the Allied forces, and then honeybee wax was actually used for waterproofing and bullet lubrication for the war. So in 1945, the war ends and the decline of the honeybee colonies begins. And you can see there's a pretty steady decline as the need for honeybees decreased in the US. And then, of course, we know in the 1980s, the parasitic mite Varroa destructor was introduced, and that kind of set off another big decline in honeybee population. So the population had kind of stabilized the number of colonies, and that introduction of Varroa set off another big decline. And if you look, starting in about 2008, the colony number in the US has actually been starting to increase. I just saw another article come out of Maine where there's a big increase in colony number in Maine. And so there's several different reasons for this. I think there's definitely been increased media attention, awareness of the general public, what's going on. And then for honeybee research, there's been a growing amount of funding, or more funding available to conduct research. But just because the honeybee numbers, the honeybee colonies are increasing doesn't mean that honeybees are doing okay. So even though the colony number is increasing, that has a lot to do with just the number of people that are keeping bees and showing an interest in beekeeping. But honeybee health is still not doing so well. So it's still on the decline. And this is a graph from the Bee Informed Partnership that's done surveys starting in about 2006 of beekeepers across the nation. And they have shown that generally the acceptable winter loss of honeybees by beekeepers is about 15%. And unfortunately, it looks like beekeepers are getting used to losing colonies because that acceptable amount of loss, you can see in gray, is starting to go up. But we're still seeing really much higher losses than that in reality. So over 35% winter loss in the 2018-2019 season. We're just seeing incredibly high losses. So honeybee health is still a big problem. There's lots of different reasons that honeybee health is in peril. So there are natural parasites and diseases. There's poor management practices. So for example, you have these large 18-wheelers taking the hives all over the country to these major commercial monocultural sites for pollination. Good example of that is the almond trees in California. Of course, when honeybees reach those monocultures, they don't have usually great nutrition there. It can also be true for our own honeybees. Depending where we live, what they have access to, there's just not as much natural forage available. So there's not as good a nutrition for honeybees. And then there are pesticides, both pesticides that the bees are experiencing, at the flower, but also the ones that we're putting in to keep those nasty mites in check. So these are all really important and really harmful to honeybees. My focus, the focus of my work, is primarily related to Varroa destructor and related diseases. So I wanted to tell you a little bit more background about Varroa destructor. Varroa is a physical burden, so it's feeding off the honeybee itself, but it's also really harmful because it's in its body, it's actually amplifying viruses. So it's not just a vector of viruses like mosquitoes can be for us, where it's spreading the virus from one bee to another, but it's also acquiring the virus in its body, amplifying the amount of virus there, and then passing that amplified amount on. So it's really very harmful. This, of course, is a picture of deformed wing virus. So you can see the deformed wings here. But you can have a lot of deformed wing virus in your colony without seeing deformed wings. You only really see the deformed wings if the virus level is very high in an individual. So Varroa destructor can harm the bee in two different life stages. It's found on the phoretic stage, where it's found on the adults, and of course it's found on the reproductive stage inside the honeybee cell. So the way the foundress works, she enters a cell. So this is your egg all the way to emerging adult here. So the foundress enters the cell right before capping. She lays a single male egg at about 70 hours. And then after that, she lays only females. And the females, as they come into maturity, they actually mate with their brother, that male, and the male will actually never leave this cell. His duty is done. But he will mate with each of the females as they reach maturity. And by the time the adult bee is emerging, she can emerge with as many as 3 or 4 mature females. So you can see how the population of Varroa can build up very quickly. That's why we don't see a lot in the spring, but by the fall we have a lot of varroa in our colonies. One of the major problems with varroa is that honeybees are quite vulnerable to varroa and other diseases as well. So they have, relative to other species like fruit flies, other insects, they have very few immune genes. So they don't have as many genes that are taking care of immune functions in their bodies. They also have a high genetic relatedness. So they're all sisters, at least half sisters. Many of them, and they're have a very high contact rate. So this is all something with, I think, coronavirus, we're all aware, if we weren't before, that this is not a good situation if you don't want to spread disease. And to deal with those shortcomings of the colony, they've developed or evolved many different mechanisms of social immunity. And I just find these fascinating. They're really cool. Grooming is one, so they can remove, this is a picture of allo grooming, where this nestmate is trying to remove this varroa from her sister. You also have auto grooming of themselves. There's something called brood comb fever, which works in a similar way to fever in our body, where the bees actually will vibrate their bodies and generate heat like they do over the winter, and raise the temperature of a brood comb and kill pathogens that way. Of course, we know now that propolis, even though it can be a pain to deal with as a beekeeper, is really, really important for honeybee health. So it has all kinds of anti-pathogenic properties. It's killing bacteria in the colony, for example. And it can do this even with volatiles. So even the scents that are coming off of it, it doesn't have to directly touch something to a little piece of propolis, but it's sending out fumes basically through the colony that are making the colony, reducing the pathogen load of the colony. And then finally, we have hygienic behavior, which is the topic that I focus on. And I'm going to show a little video of hygienic behavior. I don't know if you've seen it in your hives, but if you watch here in the middle, you'll see a brood being taken out. This is essentially the ability to detect, uncap, and remove unhealthy bees from the colony. So there they go, they're taking that brood out. There's another one coming here on the right, you can see. And so this behavior exists in all colonies to some degree. But because it exists naturally, we can selectively breed for this behavior. So in the same way that you can breed a dog to have a certain trait, like a hound dog to be better at smelling, you can breed a honeybee to be better at performing hygienic behavior. And this has actually been done for several decades now. One of the pioneers of this work is a woman named Marla Spivak, who was awarded the MacArthur Genius Grant award in 2010 for this work. And basically what she does is she selects for hygienic behavior using something called the freeze-kill brood assay. So she takes a piece of PVC pipe and puts it on the comb. She pours liquid nitrogen on top of these cells, freeze-killing just the brood in this small section of cell. Puts that back into the colony and pulls it back out 24 hours later, and then counts how many of those frozen brood the bees are able to remove within that 24-hour period. So what Marla's found is hygienic colonies remove 95% or more of that frozen brood within about 24 hours, whereas non-hygienic colonies remove less than 95%. And we found that it does work, at least partially. So we see increased mite removal in hygienic colonies compared to non-hygienic colonies. And it's really one of the most sustainable options, if not the most sustainable options for dealing with mites, because mites evolve resistance to a lot of the chemical treatments that we use. So there are several different hygienic breeds that have been developed. There's the Minnesota hygienic, which is the one I've already told you about. And they all have their pros and cons. There's the varroa sensitive hygienic, selected based on mite removal. And then there's, for example, a line out of the USDA called the pole line, which is based on mite non-reproduction. And so in the first example, we have really a pretty rapid assay. It takes 24 hours, but it's a little tricky to conduct. Even I don't really like working with liquid nitrogen. I always picture like dipping my fingers in and them shattering like glass or something. It's scary to work with. You can do it, but not everybody has access to it. And the hygienic bees are really only effective against mite if those colonies are really, really hygienic. So if they're removing 95% or more of that frozen brood. On the other side, we have the pole line, which you can get actually really good mite control with this breed. But the process of selection is very, very labor intensive. So you have scientists at the USDA going into up to 1,400 capped cells inspecting for mites, and not only looking for the mite, but also looking for offspring and counting reproductive success of each of those mites. So really labor intensive job. So right now there's no really user-friendly and highly effective assay against Varroa. And so solution against Varroa. And so there's a need for an easy, rapid, and precise selection assay. So when I started my work, I was really interested in hygiene. And one of the things I thought was interesting was hygiene communication. And if you think about the communication system, you have basically two participants, if you will. You have the signal emitter, which is the parasitized brood, and then the signal receiver, or the nurse bee. And, you know, when I started this work, we knew a pretty good deal about those hygienic nurse bees. For example, we knew that they had higher sensitivity of smell. And we knew that hygiene was performed by specialists, but we really didn't know much about the other side of the equation. So what is it that they're smelling? People had a lot of theories about what it could be. Are they smelling the mite itself? Are they smelling pathogens? But we really didn't know. So the first question I wanted to ask was, does brood really contribute to hygienic behavior? So I did this, I think, pretty neat experiment where I took brood, I took queens from different stocks. I had VSH, hygienic, this is like the pole line, the first version of the pole line, hygienic, and some unselected colonies. And I caged queens on a brood, on an entire frame. And once she had laid eggs on those frames, I cut the frames into thirds and I kind of regrafted them back together. So what I got in the end was one frame that had eggs from 3 different types of queen on it. And then I could put those frames into a new colony. When the time was right before capping of that those growing brood, I would pull them back out and actually introduce mites into individual cells, and I knew which cells those were. And then I could look back later on and look at which of those mite introduced cells had been removed or targeted by hygiene and which had not. And what I found was really interesting, and essentially what it was is it didn't matter so much where I put that frame that had the 3 different types of brood on it. It didn't matter as much where I put it, into what colony I put it, but it mattered which section of brood, where that brood originated from. So how hygienic was the colony, was the queen that brood originated from. So what that showed us is that there seems to be some sort of brood signaling that's involved in hygiene. But at that point, it really wasn't well characterized. We know that chemical signaling is really important for communication, coordination of all types of complex behaviors in social insects. So everything from family membership to territoriality, mating, for example, in cockroaches, resource quality and location, And finally, necrophoresis, or the removal of dead individuals. And this last term was coined by E.O. Wilson, the famous biologist. And basically what Wilson noticed is that you could have a dead ant in a colony, and it would be laying there clearly dead, legs up in the air, and all the other ants would just walk right on by for about 48 hours. And after about 48 hours, the ants would start to notice those dead ants, the live ants, and they would pick them up and carry them out of the colony. So he thought that was really interesting, and later went on to show that there was a chemical called oleic acid associated with that, that removal of corpses. Well, it turns out that that same chemical, oleic acid, triggers hygienic behavior in honeybees. A paper came out that showed that. But you can imagine that the signal sent out by a dead brood is very, very different than the signal that's sent out by a parasitized brood. So whereas a dead brood, like that ant that's been sitting there for 48 hours, is probably sending out a pretty strong, stinky smell, a parasitized brood, you know, a brood that's still living and maybe even very healthy at the moment, but that has a mite on it, if they're being hygienically targeted, it's with a much more subtle signal. So it turns out that honeybee cuticles, or the surface of the honeybee brood, is dominated by something called a hydrocarbon. And those hydrocarbons have two main purposes. One is desiccation resistance. So just like small babies get hot and cold really easily, or need a lot, you know, they have a high surface area to volume ratio, basically. Insects, because they're small, have the same problem. So they can dry out really easily. And that kind of waxy coating helps keep them from drying out too quickly. The other purpose that those hydrocarbons serve is communication. So I wanted to know whether these CHCs, we'll call them for short because cuticular hydrocarbons is a mouthful. So I wanted to know, do these CHCs from stress brood trigger hygienic behavior? And if they do, which ones are responsible for triggering hygienic behavior? So to find out, I took some capped brood. I, again, infested some with mites. I pulled those mites off, and I had an infested brood without a mite anymore, and then a control brood. And I extracted all those CHCs with a solvent called hexane. Basically, that's just pulling those CHCs off. And then I did two things with those extracts. I did an assay testing, basically putting them onto healthy cells to see if I could induce hygienic behavior. And then I analyzed them with something called gas chromatography mass spectrometry, which basically just tells me what chemicals are present and how much is there. So in the first case, I took those extracts that I'd pulled off of mite-infested brood, put them onto a healthy brood cell, just basically put it right onto the cap, and did the same with control brood extract. And I saw that the ones coming from the mite-infested brood were more likely to be targeted for uncapping behavior. In the second case, this is what GCMS output looks like. Basically, each of these lines, each of these peaks is a different chemical that's found on the surface of a developing honeybee brood, and the size of each peak tells us how much of that chemical is present. And what we found is that there were 10 different hydrocarbons that were elevated in unhealthy bees. In some bees, mites were responsible for driving up the amount of that chemical, and in other bees, virus level was responsible for driving up the amount of that chemical. But I'm just giving one example. Z10C33 is one chemical that we found that was elevated in response to those honeybee stressors. And this is pretty cool because it's consistent with some previous findings related to ants. So ants have a behavior that's similar to hygiene called unpacking. And this same chemical, Z10C33, has also been associated with the unpacking behavior of unhealthy ant brood in an ant colony. And we also see that there's previous studies that have linked chemicals of a similar shape and size, well, a different size actually, so much longer, but a different, similar shape and makeup to the one that we found. So basically, to make a long story short, what we found were 4 chemicals really similar in structure and all linked to varroa or hygienic behavior. And what we've done essentially with this is we think come up with a new model of how hygienic communication works. So we have 2 smaller chemicals, which are actually pretty volatile, so they can easily evaporate into the air. And we think that these 2 smaller chemicals attract specialists to the general area where there's a problem. And then those larger chemicals, the non-volatiles, help specialists pinpoint the exact problem, because of course we don't want to be removing the healthy brood accidentally. So I wanted to know next, can these alkenes be used to measure varroa resistance? So this is where we get to the tool that we're developing. It's something that we've named the Unhealthy Brood Odor Assay, or the UBO assay, because we're using those chemicals that I just told you about to essentially induce hygiene. And so we had these chemicals synthesized. We don't want to be pulling them off the brood all the time, but we can actually make the exact same chemical that we found naturally in the honeybee. We can make it in the lab. We take a mixture of these 4 chemicals and we apply it by spraying to the brood frame. And then we can measure any uncapping and removal at 2 hours. So it's a much faster assay than, for example, the freeze kill brood assay. So I want to show you an example of what it looks like. So here you have on the left, I'll show you the hygienic response, and on the right, the non-hygienic response. So we have time 0. This is just some frames I've just pulled out of the colony. We apply just half an ml, so a very small amount of this liquid to the caps of these brood, and then 2 hours later we pull it back out of the hive and we see something like this. So the hygienic response on the left and the non-hygienic response on the right. So you can get a good idea of a colony's hygienic response. We ran some tests of many different colonies, also looking at freeze kill brood assay. We tested the mite infestation levels of these colonies using powdered sugar or alcohol rinse and calculated the mite number per 100 bees. And what we found is the colonies that scored high on this assay, this UBO assay, and by high I mean they got above 60%, is what they targeted for hygiene within that 2 hours. We saw that in August, those colonies that scored high had an average of 1.6 mites per 100 bees, whereas the colonies that scored low on the assay had 6.3 mites per 100 bees on average. In comparison, the freeze kill brood assay, you see 2.8 mites per 100 bees on the high scoring colonies and 5.9 on the low scoring colonies. And this doesn't seem like a big difference, but it's almost 2 times as many on the high FKB. So basically what we've shown is that we can identify those colonies that are really good at hygienic behavior. We know that a lot of different factors may affect mite infestation levels. So we also wanted to carefully look at removal ability. So I actually introduced mites again into individual cells and then looked at how good the colonies were at removing those mites after 8 days. And again, with the colonies that scored high on the UBO assay, we saw much higher average mite removal than the colonies that scored low on the UBO assay. And then finally, and maybe the most exciting part is this assay can also be used to predict overwintering. So colonies that scored high on the UBO assay, again above 60% hygienic behavior, on average were 69% of those high scoring colonies survived the winter. And this is my research colonies, so this is absolutely no Varroa treatments whatsoever. And we had 69% survive. So the importance of this, I think, is that what we've developed, or what we're trying to develop, is something not only that works, but that's also inexpensive and really beekeeper friendly. So the idea is to put a tool into the hands of beekeepers so that they can make their own management decisions. So for example, you can say, okay, I want to treat as few colonies as possible. These are the colonies that I need to treat because they're not hygienic, and these are the colonies that I don't need to treat. Or, you know, if you have a few more colonies, say you have 50 colonies, or you want to have 50 colonies, you can take 2 or 3 queens from several different breeders and then test those queens and then choose which breeder you want to buy the rest of your queens from. So I really like the idea that we can put the tool into beekeepers' hands. The other thing that I think this tool is important for is selective breeding of hygienic honeybees. So we're using, instead of, you know, something that you hope you never actually find in the colony, which is frozen brood, we're using an actual honeybee stress signal to select for bees that can recognize that signal and respond. And what this gives us is better hygiene resolution. So with the freeze kill brood assay, you have really a narrow window of hygienic response, and that's much larger with the UBO assay. And so what we can do in terms of selection is at the lower end, maybe that's still an insufficient number of Varroa infested brood that are removed. On the higher end, you want to eliminate, I've actually seen almost like pathogenically hygienic colonies that remove even the healthy brood. They're just kind of remove everything in an obsessive compulsive way. You don't want to breed for that. But with this kind of better hygiene resolution, we can select those in the middle that are really the ones, the queens and the colonies that we want to select for, to have colonies that are better able to take care of themselves. So when we have improved management, we have improved hygienic selection, we ideally will have improved honeybee health. And because of that, we should have better crop and natural ecosystem pollination. So with that, I want to switch now to kind of the second part of my talk. Sometimes I think as a scientist, you look really closely, you have a plan, you know what you're trying to do, and you, if you get lucky, you discover something interesting. But it also happens that sometimes something interesting discovers you. And so the second part of my talk, I guess I kind of see it that way. It kind of discovered me. But if you remember back to the beginning, I was talking about splitting those frames for that, that first study. And it was really hard. So we were using, in the lab, we used plastic foundation. And I was out there, you know, 90 degrees, sweating, trying to saw these frames apart. I got frustrated. I tried heating up some wire to burn it. Just wasn't working. I didn't have the right tools. So what I decided to do was switch to wax foundation. And so, you know, this was fine, not a big deal, until a couple weeks later, I started seeing something pretty clear, I thought, in my colonies. And I usually like to ask in, you know, when coronavirus is not keeping us all on Zoom, how many people have seen something like this in their hives. So I'd love to know after I finish. But I started to notice this kind of strange occurrence related to the wires in my colony. Well, I know that honeybees are really susceptible to stress during development. So they're susceptible to things like pesticides, to poor nutrition, to trace metals. And that's not really surprising. So in just a matter of weeks, the honeybee gains 1,000 times its weight, moving from egg to pupae. So high metabolism, you can imagine that there's so many changes going on that if there's anything that goes wrong, any stress during that developmental period, it can be really problematic. We know that stress impedes population growth, and that overwintering loss is negatively correlated with colony population. What I mean by that is the small colonies are not as likely to survive as the larger colonies. You want to have that bigger, healthier population of honeybees. So looking at this, the frames like this, I thought, well, it looks like we're losing bees. And if the population of the colony is affected by these metal wires, it could be really important to overwintering success. So I started looking through the literature to find what was there, and I could only find one paper anywhere that even mentioned an effect of those wires. And what it said was there was, in honey collected from cells along those wires, there was 4.2 times more iron in that honey than in honey collected in cells adjacent to the wire. So I thought, well, what is the effect of the wire on honeybee brood? Nobody's asked that yet. And so I tested the effects of that steel foundation wire on the chemical content of the brood and also brood removal rates. I collected, in the first case, larvae along the wire. So here I have this little gray bar representing the wire. And then larvae that was right next door, adjacent to that larvae, but not overlapping the wire. And what I saw was that brood along the wire contained more iron than brood adjacent to the wire. When I looked at removal of brood, I counted the larvae along the wire again and adjacent to the wire. Then I stuck that frame back into the colony for 1 week. And then I came back and counted the larvae remaining along the wire and adjacent to the wire. And I found that the wire was actually associated with a 6.4-fold increase in removal of brood. And you can see that pretty clearly from the pictures. But what I also noticed was, I was kind of giving them the benefit of the doubt that they're building their comb right on top of this wire. But that's not always the case. Sometimes you actually have an offset of the wire where it's running through about twice as many of those cells. And so you can kind of see that if you look carefully here. For example, you have an offset wire here. Here at the bottom, you have a wire that's lined up with the middle of the cell. But based on some counting I did, which took a while, you can write this down so you don't have to do it yourself if you ever need it. There's about 3,350 cells on one side of a deep frame, which means about somewhere between 200 and 400 cells are overlapping the wire. So that's 6 to 12% of your developing brood overlapping a wire. So I think the importance of this, this study and just quantifying this is it shows that there's really, with these metal wires, a significant reduction in brood survival. 6 to 12% of the brood are exposed. You have wasted resources. So there's, of course, food required for rearing all these brood, and then they end up getting removed. So there's the time and energy that goes into removing all those brood. You also have to think about compromised worker health during removal. So of course, some of the bees are cannibalistic when they're removing honeybees. They're sometimes piercing the brood and sucking the hemolymph. And so if those honeybees, those adults, are exposed to iron, that could be a really big problem. There was a study from 2003 that showed that there are iron nanoparticles naturally in the honeybee. Of course, that's a very small amount of iron. And they thought that these iron nanoparticles are actually really important to navigation, honeybee navigation. And so if we're starting to put a bunch of iron into into adult honeybees, that could be really problematic for navigation. So to summarize kind of everything that I've gone through today, honeybees are important. They're important for crops as well as natural ecosystems. Honeybee health is declining, and varroa is a key factor, probably biologically most important factor related to declining honeybee health. Honeybee brood can trigger hygienic behavior using chemical signaling. I showed you that the UBO assay that we've developed can predict mite resistance and can predict overwintering success of honeybees, and that steel wire stabilizers can be harmful to honeybees. So moving forward, we have generated actually a lot of interest with our work with the UBO assay. We are finishing up tests in Minnesota and North Carolina. We have some planned for Louisiana and France. Have some preliminary work done in Israel, Australia, and Canada are in the plans for next summer. So lots of groups are interested in testing this assay in the field, and we hope to expand our work to not just look at mite and disease loads, but whether we can use this assay to really select for overall healthy colonies. So we don't want to select for a hygienic colony that's small and doesn't bring any honey in. We want to make sure that we can select for a colony that's really healthy overall. We are working to bring this assay to the market. So we have 2 patents that we've filed. And this is all basically owned by the University of North Carolina, Greensboro. But we are working with a couple chemical companies that are interested in commercializing this product. And Phoebe and I may be embarking in an NSF venture to try and bring this more quickly to beekeepers around the world. And just moving forward, I wanted to tell you briefly about another thing that we're really interested in is we noticed in the past 2 years testing these assays, the UBO assay, that every time we ran 2 tests within the same short period of time, the colonies always responded more high. And so we're interested in looking at whether there's some sort of actual priming effect where once the bees get exposed to these disease brood odors, they actually kind of something clicks on in their head. They say, oh, there's a problem with varroa in the colony, and maybe more bees are recruited into that hygienic workforce. And so we've written a grant that we hope to get funded to look at that next summer. I clearly didn't do all of this work by myself. There's tons of people who have been involved, including Olaf Ruppel at the University of North Carolina, Greensboro. And UNCG has been wonderful through this process, along with Triad Bee Supply and Trinity has provided equipment. Project Apis M has provided funding, and the USDA as well. So with that, let me stop sharing my screen and see if anybody's still here. And if you have any burning questions, maybe we can do some questions now, or we can jump into Phoebe's giving the Chatham County survey results. But if anybody has a question about, I know that was a lot.
I have a quick question.
Sure.
When you were looking at these frames that had the bees missing along the wires, did you see that on both sides of the frame, or was it typically just on one side?
Yeah, I did. I saw it on both sides of the frame. I still have a lot of questions actually about that work. So I don't know how, those were brand new frames. So I don't know, is that effect something that dissipates over time? Or is that something that's kind of, stays stable over time, that those brood are always removed. I think the latest recommendation is to, replace your wax every 2 to 3 years now, I think is the recommendation. So if you're replacing it every 2 years, even if it only happens the first couple go-arounds, that might have still a pretty big effect on your brood population or your bee population. But yeah, I did see it on both sides.
Had a quick comment. I think that was the most impressive bee presentation I've ever seen. That was fantastic. Very,
Thanks.
very educational. Another question about the wires. I noticed in the photograph that you used, those larvae looked almost discolored, the ones along the line with the wires. They almost had a bluish tint. Did you notice that, or was that
Definitely.
just a photograph, I wonder?
Nope, that I noticed that to me it looked like a grayish tint, and I'm still not sure if it was actually iron or metal leaching directly into their bodies, or if it was kind of a necrosis, because when they have an injury as larvae or pupae, they do start to turn kind of a grayish black. So I'm not actually sure which it was, but I believe it was actually the iron going into their bodies. And the reason is I could see it moving up the center. They kind of have a center line, and I could see it moving up the center line. So I think it was like an internal uptake of that iron. Either way, not healthy.
I can also add that I had also noticed that exact same, when you showed that frame with the clear lines, I had honestly seen the same thing before, but obviously never put it together. So, great.
Yeah, all I did was count them. That's basically all I did. But yeah, I tend to hear people have seen it. And when I was looking at, how I didn't know how common is it that people use wax frames, that people use wire foundation. And at the time I was getting them from Brushy Mountain, and Brushy Mountain said on their website that that was the #1 product that they sold, was wire foundation, wire stabilized wax foundation. And I know Brushy Mountain has closed since then. I don't know how much people are using it, but I think it's pretty common to use. And I don't have a great recommendation for alternative wax frames with stabilizers. Although I've presented this to a couple other beekeeping clubs, and I've had interesting suggestions, like maybe you could use fishing wire or some kind of piece of wood to help stabilize the wax. But I haven't tried them myself. Usually I do foundationless.
So we also had a question from David, who asked, did you notice any difference with the kind of wire? Stainless steel is less reactive and might have less effect.
Yeah, so these were, according to the company that I bought them from, stainless steel. But I didn't test that, and I didn't try any other types.
Very early in your presentation, you mentioned that bees did not pollinate other animal pollinated plants at a very high percentage, I believe. What can you give me an example of what you mean by animal pollinated, or does that include insects as well? Or
Yeah, that includes insects. So it was basically just saying that 50% of crops that rely on animals, including insects, for pollination, honeybees never even went to. So they were really important for 13% of those natural ecosystem flowers, but they went to as many as 50%. But that still leaves, basically what I'm saying is that there's 50% of plants that honeybees, that require some kind of animal for their own pollination, but the honeybees will never go to. Does that make sense? I hope that clarified it.
I've got one more question.
Sure.
Uh, are there examples of Varroa being bred in queen cells? I've never heard of that. I'm curious if in your research you've ever seen that or if you know of that.
You mean Varroa that are just naturally reproducing in queen cells?
Yes, right.
I wouldn't be surprised if it's happened, but I doubt that queen would do very well. So because that queen would emerge with a higher virus load, very likely than a sister queen that's being bred in in an adjacent queen cell, I would imagine that she loses the queen fight. And they've also shown, not with queens, but with workers, that just having a varroa mite on them reduces their weight. It makes sense. It's a pretty big parasite compared to the bee. So it's going to reduce their weight and probably their strength. So I would guess that queen wouldn't make it. All right, well, let me go ahead and share my screen again, and we'll show you the results of the survey. I'll let Phoebe take over, and then we can have a little bit more time for questions on this and Phoebe's section afterwards. So.
Sounds good. Yeah, I'll just let you know then when to advance the slides then so we can move through it. So thank you guys again for, we're really surprised. We got 33 respondents, which was great information for us and really helpful as we work to move Kaira's invention to the market. So moving on to the first question, get this out of the way here. So we asked, what are your main motivations for beekeeping? What gets you out of the house and into your hives? And so the top one was learning and curiosity, followed closely by honey production. And so some interesting answers then in the other section is that people are just interested in relating back earlier to Kaira's talk is bolstering overall hive health and keeping healthy honeybees. And the next one is supporting the environment as well as other native pollinators, which I thought was a really interesting answer. And my personal favorite was, I don't like my neighbors. And whoever said that is just a gem of a person, and I want to be your friend. I really like that one. And so for the second question, then we asked how often you're in your hives inspecting them on average through the season. And it's probably more likely that this number fluctuates through the season, but we hit about an average of about every 2 weeks. And also, we also asked how many hives do you currently have? And so the average number of the 33 respondents was 6.5. And so for the next question, we asked, what are your 3 biggest threats to your bees? And nearly everybody said varroa mites, as well as some other issues too, overwintering and queen quality, and everything else seems sort of background with pesticides, nutrition, swarming, and other pests as well. So then we asked what you use to treat for varroa in this season, either what you've already used or what you're planning to use. And about half of you said oxalic acid and Formic Pro, as well as Apiguard was in there, Apivar, brood break and queen isolation. And then for the other category, a lot of you also said heat treatment. So in the next survey, we'll have to include that in there, as that was a pretty popular one. And so with the treatment that you're sticking with, what is that costing you per hive per year? And maybe some of you had to get out some of your books and do some looking back through that to come up with a range that you're spending. So the most popular answer was $10 to $20 per hive, and less than $5, $5 to $10, and more than 20 was about the same for respondents. And then so for the next question, how effective do you feel this Varroa treatment regimen is that you're sticking with? And so it was pretty close, somewhere in the middle, very effective, somewhat effective. And so then the next question on the same slide here, how concerned are you about chemical residues from Varroa treatments in your hive and hive products? And most people said that answered this question, not at all concerned. And then there was some, a little bit of a range then ranging between extremely concerned, very concerned, and somewhat concerned. So then question 8, which of the following best describes your preferences for the use of natural Varroa treatments? So we gave you a working definition of that. So either maybe you're using hygienic stocks or formic and oxalic acid drone brood removal versus miticides, which we're calling the chemical ones, apivar strips, apistan, and checkmite. And so it seems that most people are using natural and prefer natural, but will sometimes use miticides, and it seems that miticides seem to be less popular in your county. So then moving on to the last question then, we asked about your opinion and your experience with the selected hygienic stocks, either Minnesota Hygienics, Varroa Sensitive, and the Pole line. And a lot of people have had them before and liked them. I was almost sort of surprised that nobody said that they have had them and don't like them, at least. And then another option that a lot of people responded to is that they haven't had them yet, but are interested in them. And then for the other section, there were quite a few comments on this one, is that it seems for the people that have had them have seen mixed results. So some, they've said, have seen a lot of hygienic behavior and done well with their mite counts, and others haven't performed up to par with what they were expecting. And then another interesting one was the challenge of maintaining the stocked yard. So you buy your special queen or the nukes or whatever you're buying, and then the sort of the challenge of maintaining that if your queen, something happens to her, then sort of you lose those genetics. So this gave us a lot of great information, and we were really happy to have so many people be willing to participate in this and give us this different perspective that we were looking for. So we really, really appreciate your time on this and helping us move forward. So thank you guys for participating in that. And hopefully that was helpful also, giving you some things to think about too, looking at that.
Yeah, thanks, Phoebe. And thank you everybody who participated. We really appreciate it.
Thank you guys so much. It was a great topic and I just learned a lot. I'm just still kind of overwhelmed by everything, but it was really super informative. Does anybody else have any questions? Well, I guess not.
Okay, and Julia has my email, so if anybody comes up with a burning question later, or you think of something that you didn't understand, feel free to shoot me an email, no problem.
Very good. Well, thank you again. It was certainly an educational talk tonight. So thank you guys both for doing that.
Absolutely. Thank you guys for having us.
Yep, thanks for having us back, Julia. It was great.
Yeah, we'll plan on doing it again in 2021.
Maybe in person.
In person.
Yeah, that would be great.
Yeah.
That would be wonderful. All right.
All right.
Thank you all.
Thank you.
Happy beekeeping.
Bye, everybody.
Bye.